From Microbe TV, this is TWiEVO, This Week in Evolution, episode 26, recorded on December 5th, 2017. I'm Vincent Racaniello, and you are listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Utah, Nels Elde.
Hi there, Vincent. Good to be with you from the wintry Elde Lab studios.
Did you get some snow down on the flats yet?
A little bit. So we had two storm fronts move through here in the last few days. The first one put about a foot and a half up in the mountains, maybe an inch or two in the valley, in the flats. Which is actually pretty good. The second storm front was not quite as auspicious. That was a visit from Donald Trump was here yesterday, rescinding part of the federal protections on two major areas of wilderness in southern Utah, the Bears Ears and the Grand Staircase Escalante area. So
he had
the first storm was
to visit to do that, huh?
Just to kind of rub it in. The first storm was quite good, and the second storm we're still recovering from and not quite as happy about, I would say, in general.
Nels, why don't you bring in our guest, and then we could talk about that a little bit more.
Yeah, good idea. So I'm really excited for us to be hosting or bringing in Vinny Lynch, who is a science buddy and colleague at the University of Chicago. He's an assistant professor there in the Department of Human Genetics, the Department of Organismal Biology and Anatomy. And so thanks for joining us, Vinny.
Oh, always happy to talk science.
So this Utah thing, it's opening up like parklands, right, for exploration of some sort?
Yeah, so I guess we've kind of veered from science to politics in the first 30 seconds here, but fair enough. We'll circle back.
I'm just curious the way.
No, absolutely. So. Yeah, it's actually, when you kind of dig into this issue, it gets a bit complicated. But so the basic idea here was that back in the mid-90s, Bill Clinton set aside hundreds of thousands of acres in southern Utah under federal sort of protection or oversight. And then just last year, Barack Obama did a similar thing to the Bears Ears wilderness. This was in collaboration with 5 of the Native American tribes who are in a large part living on that land. And so again, to gain federal protections. And so the basic argument is over basically who should be in control of making decisions here. Should it be at the federal level or a more local level? And so whereas it does get a bit complicated, I think one of the themes that generally emerges from this is that a lot of, at least the current folks who are in positions as sort of land commissioners and so forth, their sort of ideas about developing, oil, gas, other energy sources, including coal in some cases, are sort of what they're perhaps focused on, in addition to some of the other land uses that we might consider, including recreation. And so it's a pretty big divide between sort of the outdoor retailers who, like Patagonia, who's come out very strongly against this. They have sort of a stake in the argument because the idea being that if these lands are preserved for recreational purposes, that influences their bottom line. Whereas on the other side, you have folks who are, ranchers who actually already do have quite a bit of access to this land, but then more of the speculators who want to think about energy, and in a lot of cases, fossil fuel based energy, where that starts to get under the fingernails or under the skin of some people.
Is it possible that at the state level, the state could come out and say this is protected land?
Oh yeah, and just to say, so the deeper you go, the more complex it gets. And so it's not to say that the local people are, let's just completely open everything up. I mean, there is recognition that that as recreational land, that there's something here. But then, as you kind of look at the issues and try to see why federal kind of oversight or protection might be warranted. For example, in the Grand Staircase, it is chocked full of a lot of archaeologically relevant materials. So things like new dinosaur species, a lot of research possibilities that could be maybe undervalued in some cases or lost, depending on how you're prioritizing the use of that land. And so it's not simple, but the idea that we would be sort of leasing this off, or even in some cases potentially selling it at the more local level, and putting restrictions on public access that way is sort of a double-edged sword, or could head into a kind of irrevocable situation as well.
Well, good luck out there.
Yeah, thanks. Yeah, thanks. So lots of folks are kind of perked up about this now, and I think hopefully in the longer run, that as people who really care kind of about the possibility of building new things or discovering things when it comes to some of the archaeological stuff, for example, that this sort of patient and it takes more time to build something than to break something down or rescind something. And I'm hoping that in the longer term, that there's more momentum for the builders.
Yeah, I hope that people recognize that land is a precious resource, right? Mm-hmm. You don't want to destroy it all. And especially, national parks, and we should really leave those alone. But other lands too, let's not mess it all up.
Yeah, that's right. And so those 5 national parks here, they, and actually I would say not quite leave it alone altogether. So we're far behind on infrastructure kind of investments to make sure that things are happening sort of a more ecologically sustainable way. And so it's definitely a balancing act.
Yeah, the more people have access, maybe the more they appreciate it.
True. Yep. Which is again potentially a double-edged sword. If then I was just reading earlier today that as this has kind of come on the radar, been in the news, the number of people who visited Bears Ears, who visited Grand Staircase, even in the last year or two, has doubled, I think, twice and then tripled again, even in the last 6 months. And so that's great that people are sort of recognizing that these are amazing places to visit and to just be for a while. The other side of that coin, of course, is that then as that land is being, used in this way, that there can be impacts as well. And so it's really a balancing act.
Sure. All right. How do you know Vinny, by the way?
Yeah, Vinny, how do we know each other?
I'm actually not sure.
Probably ran into each other at a meeting, right?
Oh yeah, several meetings.
Yeah. I meet most people I meet these days. I met first on Twitter, and then I meet in real life.
Yeah.
So Vinny, where are you from originally?
I'm from downstate New York.
Downstate New York.
Yeah, so not New York City, but not upstate.
Like what city were you raised in?
I grew up in a town called Piermont, New York, which is right on the Hudson River, sort of where almost where the Tappan Zee Bridge is.
Lovely town. Really?
Yeah, I liked growing up there.
I went there once to eat at a restaurant right on the water there. It's lovely. Yeah.
Oh, yeah, probably Cornettos.
Yeah, good place. Wow. Where'd you go to college?
SUNY Albany.
Wow. You're a New York guy, huh?
Yeah.
Now, did you go as a science major?
So I knew that I wanted to do science at first I thought I wanted to fly fighter jets, and then I realized that I don't like taking orders, and I don't want to kill anyone. So that sort of nixed to that career, and then I always loved nature, so biology seemed a natural place to go.
Did you have any science in your family?
No, all my family are mostly in the military. They work for the power company as linemen or cooks. Mm-hmm. I'm the first person in my family to go to college, let alone get a PhD.
And why did you go to Albany?
Well, they had a huge program, right? So I wasn't really sure what I wanted to do. I thought that I wanted to do science. So I figured I'd go to a big school that had lots of options. There's different kinds of majors and minors to sort of figure it out in process.
Did you end up working in labs there?
Yeah, I did. So I almost immediately got into Carol Beth Stewart's lab, working on sperm protein evolution.
Wow. And then when you graduated, you went right to grad school?
Yeah, that's right. This gap year thing didn't exist then.
Yes, a lot of things didn't exist.
So then at that stage, were you already convinced that you were going to work on evolutionary questions?
Yeah, totally. So being in Beth's lab, every question was an evolutionary question, and they just seemed like the most important ones. And it seemed like they had a lot of smart people thinking about these really hard questions. And I was like, well, that's the kind of people I want to be around. And the evolutionary questions I always thought were the most interesting ones.
Neat. Where'd you end up at grad school?
So I went to Yale.
Mm-hmm.
And mostly because I didn't know what I wanted to do for grad school either. I thought maybe that I wanted to work on phylogenetics and biogeography. Mm-hmm. And there were a couple labs there that were doing that. So that's mostly why I went there.
What department were you in?
Ecology and evolution.
Oh, okay. Whose lab?
Gunther Wagner, which is not phylogenetics or biogeography at all.
What did you work on in this lab?
So I worked on a family of proteins called the Hox proteins.
Mm-hmm.
They're developmental transcription factors, but they also play roles in adult tissues. And I worked on their roles in pregnancy.
Cool. Now, did you meet Vinny Nels while he was a student, or was it past that?
I think it was past that. I think it was a little before the sort of rise of the Twitter era, but yeah.
I think that's true.
Mm-hmm. Where did you do your postdoc, Vinny?
So I stayed in the exact same lab to do my postdoc.
Ah, cool. So you were there for quite a while?
Long time. 9 or 10 years, I think.
Maybe it's a better question to ask or say. You were in New Haven a long time.
I was in New Haven a very long time. But, ever since then, no pizza can compare. So,
Yeah, well, I would. So if I'm kind of trying to rack my brain, but I would guess that we met maybe kind of towards the end of your postdoc stint in the Wagner lab. I was certainly well aware of Vinny's work already. The evolution of pregnancy turns out to be super interesting for many reasons, and that was already kind of coming onto my radar screen.
Mm.
Yeah, so if I remember right, and there's no guarantee, I came to give a talk to at Utah. And we went out to dinner with Cedric and Mario, I think. So I think that's the first time we actually met in person.
Oh, that could be. Yep. That's possible.
And out of your postdoc, did you go right to Chicago?
That's right.
And how long have you been there now?
This is year 4.
Wow. Pretty young, right, Nels?
Yeah.
Well, you were about year 4 or so, or when we started TWiEVO.
When we started TWiEVO, that's true.
Yeah.
Yep.
An auspicious year.
Yeah, I told Nels, if we do this podcast, we can track you getting tenure and work for him, So
it happened. Yeah, I would recommend that to anyone. If you're in your 4th year of your podcast lab, start a podcast and you'll be on the pathway to tenure.
Wow, that's cool. All right. Well, today we want to talk about elephants, right, Nels?
Yeah, definitely. And I guess just as you were pointing out, I think pre-show, Vincent, this might be the first podcast where we've had 2 Vincents.
I think so. I can't remember.
From the entire Twix empire of podcasts?
I mean,
is that really true?
Probably. I cannot remember it. Actually, I have the easiest thing to do would be to go look at TWiEVO. Let me just quickly take a look. I do keep a guest list.
You can just do a quick search and see.
And I can do the other shows. I'm pretty sure there haven't been any Vinny, so let's see. Let's search. Oh, Vincent Munster I had on TWiEVO. Yeah, okay. So I forgot Vincent Munster. I shouldn't. Yeah. Okay, so we're not the first, but we're the first on TWiEVO for sure.
First on TWiEVO. It took us 26 episodes to get to this stage. Yep.
Yeah, so Vinny is going to be Vinny Lynch, and I'll be Vincent. Although when you said Vinny in the pre-show, I almost responded multiple times because
there you go.
It's just genetic,
This is the problem of having 2 Vinnies on a podcast.
Yeah,
that would be a fun one to have just 2 Vinnies, and then people would get confused and so forth. But
well, maybe depending on how the podcast goes, maybe I'll step aside at the conclusion, and then you two can pick up on.
Yeah, that's funny. All right, now let's launch into
Let's
this.
Do it. So I was really excited to see this preprint, excuse me, a few weeks ago. And it's a topic that's kind of near and dear to my research heart as well, which involves these gene duplication events that can have some interesting consequences. So the paper, or the preprint, is titled "A Zombie LIF Gene in Elephants is Upregulated by P53 to Induce Apoptosis in Response to DNA Damage." pretty spicy title, I would say, right out of the gates here, Vinny. So tell us, what's a zombie gene actually?
So this actually got off Twitter. So I put out a poll and zombie won. So we named it a zombie gene because normally it's not expressed. But when there's DNA damage, it gets upregulated by P53, and it's part of the process of the cell killing itself that it contributes to. So we figured that it's something that comes back from the dead to feast upon the living, so we'll call it a zombie.
It's nice. Wow. Is this in review currently, or has it been accepted somewhere?
It's in review.
Okay. So the underlying question here is, as an animal gets bigger, it should have more cancer, but it doesn't always, right?
Yeah, exactly.
And were you always interested in that, or did this come up somewhat recently?
So I've always been a bit interested in life history because, where I went to where I got my PhD, there was a lot of talk about life history traits and how they evolve. So it's always been in the back of my head. And then I realized that I could actually turn that into a research program. And that there was actually some good models out there for doing that. Well, good. There are actually models out there for doing that. So we decided to, see if we can make it work.
So this is after you got to Chicago, right?
Yeah, that's right. Okay. It actually started with a different gene. So I was giving a guest lecture on a human evolution and disease course, and I was giving the lecture on cancer evolution. And I didn't feel like talking about the evolution of tumors. So I talked about, the evolution of cancer as a selective agent and how that affects how organisms can evolve.
So tell us how, why we think that it's not just being large, but also living a long time, you might have more cancer. Why is that?
Yeah, so the logic is pretty simple, right? So if all organisms have the exact same chance of getting cancer, then the big ones, which have more cells to potentially turn cancerous, should just have higher cancer rates, right? More cells that can turn cancerous, you should have higher cancer. And you don't observe that in the world. So cancer rates in wild populations that aren't exposed to toxins and the like are more or less the same. So 3 to 10% or so, regardless if you're a whale or an elephant or a shrew. And the same logic applies for lifespan, right? So the longer you live, the more time you have to acquire a cancer-causing mutation. So things that live a really long time should just have more cancer because they should have acquired more cancer-causing mutations. But again, you don't observe any correlation between lifespan and cancer incidence either. So, this has come to be called Peto's paradox because he was one of the first people to recognize it. The resolution to the paradox is really simple, right? It's just evolution. Right? These big, long-lived things evolved to enhance cancer suppression mechanisms.
Right.
But figuring them out is hard.
But you say that within a species, though, if the larger members do have more cancers, is that right?
Yeah, they do. That's right. So if you're taller than the mean, if you're a human who's taller than the mean, you are at increased risk of cancer compared to a human who is shorter than the mean. So it's cancer protective to be short. And you can also see it in other animals. So there are actually a lot of species that there's a dramatic variation in body size. But it works for dogs, and whatever, they've been selected by us to do all sorts of things, and maybe that's increased their cancer risk. But for little dogs, they very rarely get cancer, and very, very big dogs get a whole lot of cancer. And it's a pretty strong relationship in dogs.
I'm glad that there's some good reason for me to be short.
It's cancer protective.
Although my father was shorter than I, and he died of cancer, so it's not an absolute thing.
No, there are no absolutes.
Yeah,
it's actually worse than you would imagine, right? So if you work out the math, an organism which is twice as big as as a different organism should be, just based on body size, have double the risk. But if you do the math by lifespan, it's t to the 6th power, where t is the difference in lifespan, right? So it's a lot more pronounced for lifespan than it is for body size.
Right. Now, how long have people been interested in this? Well, I guess it's been known for quite a while. When did Peto work, actually?
This is the 70s.
70s. So people have been trying to figure this out since then, right?
Yeah, but it's always been very theoretical. So people have come up with, 327 different reasons why these large-lived things have evolved enhanced cancer suppression mechanisms, but there actually hasn't been a lot of empirical study. And that's just because we didn't have the tools to do so until relatively recently, right? Until comparative genomics came online. Presumably these things are genetic, right? So you need to sequence the genomes of lots of things. And it might not always be that the tumor suppressors, which you think are the important genes in one species, are the important genes in another species. So you need sort of an unbiased way to do this.
Of course, I'm sorry, Nels, one last thing.
Go right ahead. Yeah.
Until we figured out the basis for cancers, which involved, tumor suppressors and oncogenes and all DNA damage, which is a pretty recent development, we couldn't have even start to think of mechanisms, right?
Exactly right. So it relies on having the technology to do comparative genomics, and it relies on having a relatively good understanding for the sort of basic biology of cancer.
Mm-hmm. Yeah, and so we'll get into the Lift gene, which I guess is a tumor suppressor. Is that right?
Yeah.
Yep. In a few minutes, but just to say, I guess this is not your first rodeo with Peto's paradox. You had some really interesting work just even in the last few years on P53, actually, of sort of one of the more famous genes when it comes to cancer genetics. Maybe tell us a little bit about that.
Yeah, so that we got really lucky there. So back when I was giving that guest lecture on evolution of cancer suppression mechanisms across species, I thought, well, we have genomes for elephants and their closest living relatives, which are manatees and hyraxes, and no one's ever really heard of a hyrax. But it's really small. It's about the size of a groundhog. It sort of looks like a groundhog, except it has wicked fangs. It has really large incisors like an elephant would. So we have genomes for these things. And I thought, well, let me just blast the genome and see, if you were going to pick one gene to see if elephants have more of them, you'd pick P53, right? And the logic there was that, well, elephants have enhanced cancer suppression mechanisms. Maybe they just have more tumor suppressor genes. And if you're going to pick a tumor suppressor, you pick the master tumor suppressor. And it worked. They had 20. Their closest living relatives had one or two had one normal gene or one or two pseudogenes, but elephants had 20 copies. So we got really lucky in that, the first gene I picked to blast, elephants had an increased copy number.
Yeah, very cool. So, and actually, that's an interesting that I remember you giving that talk here at Utah. And in fact, there's another Utah connection, right? Wasn't there another lab that was sort of on the same path, or kind of thinking slightly the same way at the same time? Maybe tell us a little about that story. It's kind of an interesting.
Yeah, so those are our frenemies.
Yeah, there you
go. So back when we were getting ready to publish this thing, right? So we were writing it up, and I was like, hey, I find it really remarkable that no one has noticed this before. Because I can't be the only one who just thinks, well, maybe elephants have extra copies of tumor suppressors and sees if they have extra copies of P53. But if you did a PubMed search, nothing came up. Mm-hmm. So I just started Googling around, and there was a link to an American Cancer Society talk by this guy, Josh Schiffman, who's at Utah. Mm-hmm. And I was like, the title was something along the lines of, elephants have lots of tumor suppressor genes. So I watched it because it was recorded, and it was basically him talking about all the data in my paper. So there are two options when that happens. We can either rush to publish, or I can call up Josh and say, we're doing the exact same thing. Do you want to keep each other aware of where we are in our publication timeline? So I did that. We exchanged several emails and called a couple times and let each other know where we were in the publication process. So just as their paper was about to be published in JAMA, he let us know, and I put our paper on bioRxiv.
Yeah, so this is actually when I tell this story from time to time. First of all, just because Josh is a colleague here at Utah over at the Huntsman Cancer Center. But this seems to me like actually a really good example of how the bioRxiv can sort of help mediate some of these sort of competitive scenarios that can emerge in science from time to time. So it seems in this case, because you were able to put your manuscript up on the bioRxiv, everyone could know that, hey, look, this is the sort of spot where we were in thinking about these questions, and it kind of gives it a nice timestamp for comparing the two studies.
Yeah, exactly. I mean, we could have rushed to publish, a year before we actually put the thing on bioRxiv, but there was this one crucial bit of data that we really, really wanted and knew that we could get if we could just work out the experimental system a little better. So I could have rushed it and published first, but that would have totally scooped Josh and his group, and that's not a nice thing to do. I don't want to do that. And the existence of bioRxiv lets us do things like this, right? Like play friendly with each other, and you get sort of a more accurate representation of the trail of knowledge, right? You get better provenance with this kind of thing.
Yeah. And so how did it work out, Vinny, in the end? Did your paper then get published?
Yeah. So we published in eLife.
Mm-hmm. Yeah, that's great.
Vinny, if you look at the evolution of animals, can we tell when cancers first emerged, or have they always been there in the smaller ones?
So you can actually see some evidence for cancer in the fossil record. So there are lots of examples like dinosaurs that get cancer and stuff. But really, it should be a problem of multicellularity, right? Like once you have more than an organism composed of more than one cell, then it should increase the fitness of that other cell to increase its proliferation rate, which is basically what cancer is, right? As soon as you get multicellular, you're at a theoretical risk for cancer.
Do we actually see that today in small multicellular?
So we would we don't see things that we would call cancer. But you see things like clonal evolution, where you'll get one cell that seems to proliferate really rapidly. But I don't know that I would call that cancer, right? So it's something that's akin to it, but it's not quite the disease process, right? Because if you get a benign tumor, technically that's cancer, but it's not going to kill you.
Right.
But if you get a metastatic tumor or pancreatic cancer or something, that will kill you. So these are two ends of the same coin of what cancer is. So there are things like it, but it's not quite what I would call cancer.
Now, as I'm thinking of clam cancers, but that's not very small, right?
Right. But that does
they're transmissible.
Yeah, they are.
Yeah, exactly. That does kind of get a little bit at these ideas. So this was TWiEVO. I'm just looking now, episode 17 with Steve Goff.
Yeah, it's a really cool story.
Yeah. Yep.
Yeah, he's right next door to me here, Vinny.
Oh, really?
Yeah, it's my neighbor.
Yeah, we read that paper in one of our departmental journal clubs.
Yeah, Steve and I have been next to each other. He and I were both postdocs in the same lab, and then we both came here to Columbia.
It's funny how those things work.
Known him for a long time, yeah. So you had shown that the P53 gene in elephants was duplicated, is that right?
Yeah, that's right.
And then what got you on to LIF?
Yeah, so the lab was also another sort of accident. Half the lab studies sort of Petto's paradox questions, and the other half studies the evolution of pregnancy. So there are some genes that are really important for pregnancy that I just know a lot about because of these other studies. So what happened was a grad student in the lab was working on the evolution of cancer story. And what we did was identify all of the gene duplicates in elephants, hyraxes, manatees, and a couple other afrotherians. And then we identified elephant-specific gene duplications. And that gave us a list of whatever 350-something genes. So I was just staring at this list of 350-something whatever genes, and just at the top by chance was LIF. And LIF is the abbreviation for leukemia inhibitory factor, right? So it sort of says it right in the name that this might be a particularly important gene for cancer biology, but it also plays an incredibly important role in pregnancy biology. So in the pregnant uterus, P53 gets turned on by progesterone, and it turns on LIF. And you need to turn on LIF in the uterus in order to have implantation. So I just happen to know a bit about the biology of LIF. So then I started to look into what that gene was doing in a little more detail.
Wow.
That's cool. So in the P53 case, you had all of these gene duplications. So and we've talked about duplication, but maybe let's get into more of the details of that. How do these duplications actually arise in the first place?
So for P53, it's what we call a retroduplication. So this is a gene that gets transcribed into a piece of RNA, and then that RNA gets reincorporated into the genome. So when that normally happens, those genes tend to be non-functional because they lack regulatory elements like promoters and enhancers. So there's no way to turn them on. With the P53 story, elephants found a way to turn on some of those retrogenes, which normally you'd consider was a pseudogene. And with the LIF story, it was a duplication of the locus. So instead of going through that RNA phase, that bit of DNA that encodes the LIF gene was duplicated just downstream. So there are two copies in tandem almost right next to each other.
So just from reading the on the bioarchive, so there are some transposable elements, aren't there, in the neighborhood? Are those just kind of hitchhiking along, or do they have any role in the segmental duplication, or is that just totally separate?
No, I think they're mediating the segmental duplication. So if you map the ends of the duplicates, they map either within or very near to the transposable elements. And one way you get segmental duplications occurring is that during crossover and the like, you need to have pairing of the two chromatids. And if you have really repetitive regions, then you don't know which part pairs correctly. And if you get incorrect pairing, you get one copy of the chromosome having extra copies of the gene, and the other copy of the chromosome having fewer copies of the gene. So I think that's what's going on here.
Mm-hmm.
Those TEs are mediating unequal recombination between the two chromatids.
That's interesting how these sort of selfish genetic elements are having a hand in this sort of in different ways in the two stories relating to cancer.
Yeah, absolutely.
Yeah.
So you looked at 53 mammalian genomes, right, from big and small animals, right?
That's right.
And these are all genomes that are in databanks, right?
Yep. This is all stuff the government has paid for already.
And you picked them a range of sizes, I guess, big and small. That was really what drove it.
Yeah, so we tried to get as many mammals as we could and across as many different body sizes of mammals as we could.
Okay. And all mammalian genomes have at least one, right?
That's right.
And is it just no, it's not just the elephant. The big guys have more than one, right?
So elephant, manatee, and hyrax all have more than one copy,
Right.
but only elephant has an additional functional copy. So in
Right.
manatee and hyrax, all those extra copies are pseudogenes.
I put a picture of a hyrax in the notes, Nels, by the way.
Yeah, I saw that. It's a striking critter.
Wait.
They're really cute.
Yeah, a lot of the pictures are cute, except when they growl, and then they look kind of scary.
And then those incisors come into
That's right.
Yeah.
Although when you look at the rock hyrax, you wouldn't immediately say, "Oh, wow, there's a close relative of the elephant."
No, you absolutely would not. The cool thing is that there are some extinct hyraxes. So hyraxes now are very small. They're about the size of a groundhog. But there are these extinct hyraxes, like Titanohyrax, which apparently was the size of like a rhinoceros.
Oh, wow.
Mm-hmm. So, Vinny, tell us about the different kinds of lift genes and what they do. I think that our listeners need to know that.
Yeah, so there's one lift gene, and it encodes 3 different isoforms. So isoforms are just different variants of a gene, and they differ in the part of the gene that's included. So lift normally functions as a cytokine. It's responsible for signaling between cells. So a cell makes lift, and it exports it into the space between cells. And another cell has a receptor that can bind it, and that lets the other cell know that, hey, I've just received a signal from that first cell. So one of the isoforms is one of these extracellular cytokines that does signaling. The second isoform, no one is really quite sure what it's doing. And the third isoform never gets sent out of the cell. So it lacks a bit up at the start of the protein, which is responsible for packaging it for export. Mm. So because it lacks that bit of protein, it can never get exported. So whatever it does, it has an intracellular function.
And this intracellular lift is only in elephants?
So it's in everybody. It's just that in all of those other mammals, the one lift gene encodes all 3 of those isoforms. And in elephants, they have one normal lift gene that's capable of producing all 3 of those isoforms. And one of their extra copies is only capable of making the intracellular isoform.
I see. So would that increase the amount of protein that would be present?
So it increases the amount of protein by probably a lot, actually.
Wow.
And it biases, that extra copy that's making that extra protein. Mm-hmm. Since it's only able to make one of the 3 isoforms, it sort of biases the balance of all the 3 isoforms in the cell toward isoform 3.
Okay. And also there's another story here, and that's apoptosis and lift, right?
That's right. So one of the original functions for lift is that it keeps cells. It helps cells proliferate or inhibits proliferation, depending on the context. So that's a function that's performed by the first 2 isoforms.
The ones that are secreted, right? Yeah.
That's right. Yeah, but the third function of that smaller isoform, the intracellular one, is to induce apoptosis. At least one of the functions of that third isoform is to induce apoptosis, which is just programmed cell death. Once that gene gets turned on, it causes the cell to kill itself.
All right. So normally in elephant cells, this gene is off.
That's right.
Mm-hmm. And what actually turns it on to execute that cell death pathway?
So at least the elephant isoform, the extra copy in elephants, gets turned on by that tumor suppressor P53 we were talking about. So just upstream of where the gene starts, there's this beautiful P53 binding site that's sitting there. Mm-hmm. And the thing is, the gene is normally turned off. So if in a normal healthy cell, this gene is almost completely silenced. But when the cell senses DNA damage, it turns on P53, and then P53 can come in and turn on this gene.
So if P53 is required for transcription of this LIF gene, right?
That's right.
You call it LIF6, right?
Yeah, because it's the sixth one we identified in the elephant genome.
Yeah, and maybe actually why don't we circle back from it and just think about the some of the evolutionary questions. So in that initial comparison of 53 species, you show that there's only a very small subset, those three, the manatee, the hyrax, and the elephant. So what does this tell you about sort of the origin of the duplications, and how is that useful for sort of considering the function?
Yeah, so, presumably everything that's multicellular is at some risk of developing cancer, right? And, it's true for the really, really big things or the really, really small things. So if it's so easy to get enhanced cancer suppression by, duplicating your tumor suppressors, you would just expect lots of mammals to have duplicated tumor suppressors like P53 or like LIF. But we only observe a functional duplicate in elephants. So normally all these other mammals have one copy. And elephants are the only mammals that have two functional copies. So presumably there's something that's preventing those other animals from evolving more than one functional copy of LIF, right? Because it's not hard to duplicate a gene. Genes are duplicating all the time, which suggests that there is a trade-off or some constraint on having extra copies of LIF around, that we only observe it once, and that's suggesting that whatever broke that constraint is relatively rare.
Mm-hmm. Could you put this LIF gene in a mouse and see what happens?
Yeah, so we're gonna try to do something like that. We're not gonna do mice because their lifespan is relatively it's not long, but it's long for a laboratory organism, like 5-6 years. But there are various species of fish which don't live very long, and this gene is conserved in fish. So we're gonna try knocking it into some species of fish to see what the effects are.
Yeah, cool idea. Although I guess so once you identified the these extra copies or those duplicates, then the next question might be, just as you said, because there might be these constraints on kind of not having that extra function or that extra copy, is whether these things are functional at all. And so,
exactly.
Yeah. So how is it that you kind of went through that process of figuring out whether the elephant version was functional? Maybe walk us through that a little bit.
Yeah, right. So just because you have an extra copy of a gene in your genome doesn't mean that gene is functional. So we basically walk through the central dogma, right? So if that gene is functional, and genes, in order to function, need to be transcribed, well, we should see evidence of transcription for that extra copy. So what we did was generated some RNA-seq data and used some existing RNA-seq data from elephant cells and tissues and said, okay, the elephants have an extra copy of that gene. Is there any evidence that gene is transcribed? Is there any evidence it's expressed? And when you look in the RNA-seq data, you see a very tiny little bit of evidence for expression. So it's there, but it's barely there. But that sort of makes sense, right? Because when this gene gets turned on, or at least when we artificially overexpress it, it causes the cells to undergo apoptosis. So you actually don't want this gene being turned on all the time because it's going to be killing the cells all the time, right? Mm-hmm. So it sort of makes sense that it's not expressed in normal basal conditions. So then what we did is we reasoned that, well, if there's evidence for a little bit of expression, maybe the expression is inducible, particularly given that this thing causes the cell to undergo apoptosis. So we took our elephant cells in the lab and gave them lots of different kinds of drugs to induce DNA damage, which is the kind of thing that normally leads to apoptosis. Mm. And when you do that, then you see that the expression of this gene goes way up. So it gets massively turned on once there's a bit of DNA damage around, which is almost exactly what you would expect, right? So if this thing is inducing apoptosis, and apoptosis is mostly an effect of DNA damage, then there's a very nice connection between those two things.
Mm-hmm. And it was not a problem to distinguish all the isoforms from the other, from LIF1 and LIF6 by RNA-seq?
No, fortunately, these things duplicated long enough ago that they've diverged a bit in their gene sequences. So it allows us to unambiguously map in RNA-seq data.
Mm-hmm. All right, so treating with a DNA damage agent, how much does the transcript go up?
Well, it depends on the DNA damage agent and how much you give it, but you can make it go up 10 to 100-fold if you play around with these things.
And then the cells die?
Yeah, then the cells very rapidly die.
Mm-hmm. And so I guess because there's LIF6, so that implies that there's at least 6 of them. And is it the case that LIF6 is the only one that is upregulated by the treatment with DNA damage, or by using DNA damage.
Yeah, so it's the only one that we've found to be upregulated. So a couple of the other duplicates have signatures of functional genes, and by what that means is that if you sort of look to see how diverged they should be, if they're totally non-functional pseudogenes, they're not quite that diverged. They're less diverged than that, which suggests there's purifying selection acting on them. So that's an indirect way of inferring function. The problem is that we don't have RNA-seq data or RNA from a vast array of elephant tissues to go through all the tissues and say, okay, are any of them tissue specific in their expression, right? So it'd be great to get all the different kinds of cells from all the different kinds of tissues, culture them in the lab, give them DNA damage, and see which genes differentially go up and down. We can't do that because the only cells that we have in the lab are dermal fibroblasts. So all we can say is that in these dermal fibroblasts, LIF6 is the only expressed gene, which doesn't exclude that the other genes are turned on in response to DNA damage, but just in other tissues.
Mm-hmm. And what's going on actually with the ancestral or the parental copy of LIF? Is that still in the game, or is LIF6 taken over for the elephants?
It seems like LIF6 has taken over. So we can identify all three of those different isoforms for the parental copy. It's just that the third isoform, the normal one that induces apoptosis, it doesn't get upregulated that much in response to DNA damage. So it seems like maybe this one has taken over the function.
Remind me, does LIF1 have a P53 element in its promoter?
It has a P53 response element in the first intron. Okay. So it's there.
Yeah.
So how does this fit back into the Peto's paradox, I guess? Is it that LIF6 is sort of more potent as a tumor suppressor relative to the ancestral form, or is it sort of a one-for-one exchange? How are you looking at that?
So I think it's a dosage. Is just upregulated significantly more than the original isoform 3 that was able to do this, and it gets turned on so much that the effective dosage just overwhelms the original copy of the gene.
Interesting. So, and I guess you term this as refunctionalization.
Yeah, because we know it went through. So by doing some evolutionary tricks, we know that this gene went through a period of being a non-expressed pseudogene. Mm-hmm. Because we can reconstruct how the evolution of its promoter worked. And if you look at ancestral versions of its promoter, or likely ancestral versions of its promoter, the promoter is non-functional. There was a region of DNA which is close to acting as a promoter, but needs a couple mutations to evolve into a promoter. Mm-hmm. And that happened in the common ancestor of elephants and some other elephant-like lineages. So we know that it went through a period of non-functionality.
And that's where that's where zombie comes from, right?
That's where zombie comes from. It was dead.
It was dead.
But it has come back to wreak havoc.
I
like it.
Can you explain for listeners mainly why DNA damage would warrant having a tumor suppressor or an apoptosis response?
Yeah, so your cells have evolved lots of ways of repairing DNA damage. So DNA damage usually means that there's been a break in the strand of DNA. And it can either be a single strand break or a double strand break, which means you have two strands of DNA just floating in the cell. And the cells don't like that because it means that the next time they replicate, they won't be able to replicate that chromosome that has the break. So they found a way of rejoining the ends and sticking that break back together and repairing the damage. The cell has lots of ways to do that. It's just that it's error prone. Mm. So there's a chance that when you repair the damage, you'll repair the damage, but you'll introduce a mutation. And if you introduce enough mutations, then you're going to either cause a tumor suppressor, a gene that's normally going around surveilling the cell and saying, is it turning into cancer? If you make that gene non-functional, it can no longer surveil for tumors anymore, right? So DNA damage is what causes cancer. Mm. And if you acquire too much DNA damage, the probability that the cell is going to be able to correct all of it without introducing mutations is very low. So the way the cells deal with this is they have sort of an internal clock. And if they can measure how long it's taking for the genome to be repaired, And if that takes a really long time, it probably means that there's a lot of damage. And then it sets off a little stopwatch. And if it goes too long, it just kills itself.
So it's a kind of a preemptory strike, right?
Yeah, it's sort of the needs of the many outweigh the needs of the few.
Because we're saying, well, you're likely to have serious problems, so we're going to get rid of you.
Exactly. Since you're likely to have since this cell is likely to have serious problems, kill the cell and don't ever see the consequences of those problems. So
in terms of virology, this is interesting because it turns out most cells are not replicating or not duplicating all that much, and viruses don't like that. Well, I shouldn't say they don't like it. That's anthropomorphizing. But viruses don't replicate in quiescent cells. So many viruses kick cells into, dividing. And they have proteins that do that, like T antigens of SV40 and all that stuff. And then, of course, the cells pick it up. They say, wait a minute, we're not scheduling this DNA replication. And that turns on P53 and induces apoptosis. But the viruses, of course, are not going to succeed unless the cell lives. So they have evolved suppressors of apoptosis. And that's how it works. So a virus can kick on DNA replication and cell division and still keep the cells going because they're inhibiting apoptosis. And so this is a kind of a neat little example because this LIF6 induces apoptosis. And I would bet that, there are viruses that infect elephants for sure. I'm thinking of elephant herpes viruses, right? And
that one's pretty bad.
And they have got to have a way to antagonize apoptosis. Probably not at the level of LIF6, but at some other stage in apoptosis.
Yeah, I mean, it's very interesting that there are a lot of kinds of bad things that happen to the cell that seem to turn on very similar phenotypic outcomes, apoptosis, right?
Yeah.
That if the virus keeps replicating in that cell, that's not good for the organism either, right? So then, just kill that cell.
Yeah, of course, if they can, because some DNA viruses don't allow it.
Yeah, well, I think that's right. I mean, this is another area where sort of the evolutionary view on cancer or an evolutionary view on virology, I think, can become pretty interesting as well, because you have the viruses sort of vying for control of cellular cellular life, or continuing to have a virus factory, and then that sort of is entangled with some of the same mechanisms that might be at play for suppressing cancer. That you can imagine, then, that these messy evolutionary histories, when they collide like this, could have an influence on things like cancer susceptibility outside of infection as well.
Yeah,
absolutely.
Sure. Mm-hmm. You did some cool experiments with siRNAs to show the role of LIF6. Can you just summarize those for us?
Yeah, so that's walking down the central dogma a little further, right? So what we had done is shown that the gene makes an RNA, but you need to show that RNA does something functional in order for there to be a causal relationship between the duplication of that gene and it having a role in resolving Pento's paradox. So we reasoned that if expression of that gene is important, if we knocked down the expression of that gene, we should observe some phenotypic outcome. So since we know that the gene, if we overexpress it, causes apoptosis, we used an siRNA to knock down the expression of that transcript, of that gene's transcript, and then measured if that influenced how the cells responded to DNA damage. And specifically, we asked, if we knock down the transcript and give the cells drugs that induce DNA damage, do we see a reduction in apoptosis? And we did, actually. So that suggests that gene is playing a causal role in the induction of apoptosis.
Yeah, I like that. It's a nice experiment. And these are done in elephant cells, right?
Yeah, that's right. So we have African and Asian elephant cells in the lab. Mm-hmm. And also hyraxes and lots of other fun critters.
Too bad it wasn't refunctionalized in the hyrax. It's still, the zombie is still dead, I guess, in that case, right?
It is. They found a way to keep it dead.
Yeah. So then you went on and you also looked at where the protein is localized. Maybe tell us the motivation for those experiments as well.
Yeah, so there's two major pathways for the induction of apoptosis. So, for example, LIF6 could have been acting as a transcription factor, going into the nucleus and turning on genes which then lead to apoptosis. or it could be localizing to other organelles in the cell that play a role in apoptosis. And there are a couple really important organelles that play a role in apoptosis, and that's the endoplasmic reticulum and the mitochondria. And the major one is the mitochondria. So the mitochondria has an inside and an outside, and when the stuff on the inside gets to the outside, it activates this whole pathway, which leads to apoptosis. So we tagged the Lift6 protein with a green fluorescence tag, and then just looked to see if it was localizing to the nucleus, the mitochondria, the ER, or a bunch of other organelles. And we found that when we overexpress it in different animal cells, it localizes almost exclusively to the mitochondria, which makes a lot of sense, because what happens when you induce apoptosis is that p53 is a transcription factor. It goes into the genome and it turns on the expression of a bunch of genes which localize to the mitochondria and cause pores to form in the wall of the mitochondria. And those pores allow the inside of the mitochondria's contents to leak to the outside. And that's what directly activates p53. That's what directly activates apoptosis. So it looks like what Lift6 is doing is participating in the formation of those pores in the mitochondrial wall.
Another question I had, Vinny, was so when you upregulate Lift6 through p53, is that actually through the elephant's original p53, or is it through those extra copies of p53 that you were describing from before?
Yeah, so it's through the original copy of p53. So all those extra copies of p53 that elephant has, all extra 19 copies that they have, they're incomplete. They all lack the DNA binding domain. So whatever they're doing, they're not directly binding DNA, so they can't act as a transcription factor. The only version of the elephant p53 genes that can act as a transcription factor is the original copy.
Gotcha. I was kind of hoping that one of the extra retro copies might have been involved. It'd almost be this weird conversation between the selfish genes, like kind of echoing through evolutionary time somehow.
Yeah, so actually I think that they are, but they're not doing at the level of transcriptional regulation. So, in some follow-up studies to the elephant p53 work, it looks like they're also localized to the mitochondria, and that they also play a role in inducing pores in the mitochondria. So it seems like all these extra copies of things in elephants are all sort of closing in to cause the mitochondria to basically explode.
Interesting. Yeah, cool.
Could I ask you something about mitochondria and apoptosis? Would that be an appropriate question?
Of course.
So I was reading a book by Nick Lane. Do you know this fella over in the UK?
I don't.
He's written a couple. He's a very interesting guy, scientist. He's written a couple of books, kind of for the public, which are really good. I'm reading one now called Oxygen. It's all about the rise of oxygen in life and on Earth. Really interesting stuff. But I read another one about the origins of life, and he thinks all life, all life on Earth is based on proton gradients, right? And,
that's right.
He thinks that if any life evolved anywhere else, it would probably also be based on such thing. And of course, the mitochondria, it's crucial to have a proton gradient, right?
Exactly.
To generate ATP. And so he discusses how if the membranes of the mitochondria get leaky, you're going to screw up the gradient, and that's why the cell dies, to take that out. And the selection is a conflict between the nuclear genome and the mitochondrial genome, because if they're mutating, if they're evolving independently, it could arise that the proteins are not matched properly, and that's why you would kill the cell, right?
Yeah.
So do you think that is what drove the evolution of apoptosis?
So that is one of the explanations for what drove the evolution of apoptosis. And some people think that because the mitochondria originally was presumably a parasite on the eukaryotic host cell, and somehow they've evolved a way to be mutual, right? To work together for each other's best interests. But maybe the host genome has followed the trust but verify rule, and it was originally a way of keeping the mitochondria in check should it get out of control.
Yeah, and then so we know now apoptosis is a response to virus infection. So that was kind of a benefit that accrued from that process, I suppose, right?
Yeah, exactly. I mean, evolution repurposes stuff all the time, right? Like, look at these pseudogenes that evolution has repurposed. Particularly, it's going to repurpose pathways in any way it can, they pop in. So, yeah.
That's very cool. Although I think the viruses probably preceded, for sure they preceded eukaryotes, invasion of cells with bacteria. But maybe there wasn't any apoptosis back then.
No, it had to come around at some point. So
I mean, would you say that came around when the mitochondria came in, right?
Either when the mitochondria came in, or at the moment just that multicellularity evolved.
That's supposed to be coincident, right? Before the mitochondria came in, there was no multicellularity, right?
As far as I know, it's supposed to be coincident.
Yeah. Okay.
And
that's a tough one when we sort of gaze back, north of 1 billion years. That things can get a little fuzzy.
Yeah.
Oh yes.
And I think it's still the case that multicellularity evolved independently multiple times. So presumably these organisms that independently evolved it all had to find ways of dealing with the cancer problem.
Right. But they evolved independently, but they all happened after endosymbiosis with the mitochondria, right? Yeah.
Mm-hmm.
Yeah. That's what that extra energy is the kick that allowed it, I guess, right? That's the idea.
I think that's what people hypothesize.
It's pretty cool stuff. Now, this Lift 6, it's just in elephants, right?
Yes.
It's not in like whales.
Nope.
Right. So how do they solve Pita's paradox?
God, I wish I knew. So we've done the genome comparison thing.
Sorry, I said Pita. I should have said Pito. Pita is bad.
Yeah. They're causing problems at Yale. So there's this lineage of whale called the Bowhead Whale, and its genome is sequenced, and it lives a very, very long time, so at least 230 years. And it's closely related to whales, which are nearly as big, but live maybe 80 or 90 years. So we've compared the genomes of whales, including the Bowhead Whale, to see could we identify anything like LIF or anything like P53. And the answer is no, that we have not found any obvious gene duplications in those lineages that we would just be able to look at the gene and say, well, yeah, obviously duplicating that gene is going to increase cancer resistance. So however they're doing it very differently. And the really interesting thing is that most whales, which are very big, so think like sperm whales and blue whales and humpback whales and bowhead whales and right whales, they're ancestrally small-bodied whales. So large bodies have evolved independently multiple times in whales. So either the ancestral whale was particularly good at suppressing cancer, and that allowed large-bodied, long-lived ones to evolve multiple times, or each of those lineages has independently evolved cancer resistance. We don't know what it is, and I don't know that it's knowable. But they're mysterious, all right.
But you could probably you can figure out eventually why or how, what the mechanism of cancer resistance is, right?
Oh yeah, totally. If it exists, we can figure out the mechanism, right? So biology is not quite a black box, even though there are parts of it that are. So we'll figure it out.
What is the advantage of becoming large like a whale or an elephant?
So life history, so here we go back to life history. Life history theory predicts that if you grow to be very large, you decrease your extrinsic mortality, which basically means that things aren't going to prey upon you if you're very, very big, right? So whales and elephants, there's not a lot of things that prey upon them when they're full-bodied. I'd venture that other than us, probably nothing. So that should lead to a bias in the direction of evolution toward large bodies. And if you sort of look at the mammalian data, you certainly see that. There is multiple convergent increases in body size in multiple lineages. There's definitely trade-offs to evolving large bodies, right? It takes a really long time to evolve a large body, time in which you're not reproducing. So things that are very big tend to take a long time to mature. So there are trade-offs, but at least within the mammals, it seems like you decrease your extrinsic mortality.
And that's why these large animals live so long, I presume, right?
Yeah, so there's also a relationship between how big you are and your metabolic rate. So metabolic rate scales, and big things have very slow metabolisms, which maybe also allows them to live a little bit longer.
Wow.
So in this idea of, growing big to avoid predation might have worked for a while, but certainly once our species kind of got into the game, it seems like this didn't work out for a lot of the megafauna that's now extinct. Are there any prospects of going back to, other sort of archaic samples or ancient samples to try to mine the DNA and actually kind of ask the same questions about how, like maybe in independent cases, that you might solve Peto's paradox?
Yeah, so we're trying, at least in the elephant lineage, right? So we have woolly mammoth genomes, and there are at least one mastodon genome. Mastodons they look like elephants, but they're not really elephants. So that lets us push back in time when these Peto's paradox questions arose.
That's cool. Yeah, that's really cool, isn't it?
I agree. But I'm wondering, I guess the mammoths are related to the elephants. The mastodons aren't, but are there other kind of independent sampling points possible? Or are those kind of missing from the fossil record or the evolutionary record now?
So we can infer a lot about an organism based on its skeleton, right? So the skeleton leaves a pretty good fossil record. So we can reconstruct using evolutionary methods when different lineages evolve to be big. And because lifespan correlates with body size, we can infer the lifespan of those things. But that's just about where our ability to infer things stops, unless they died recently enough that we can get ancient DNA samples from them.
Yeah, it's too bad you can't get dinosaur DNA. It would be so cool to...
It would be.
Right, figure out what this situation was there. It's a long time ago, right?
I want Brontosaurus DNA. But one of my professors in college told me that you have to work with the biodiversity you have, not the biodiversity you want.
Yeah, true.
That's really good. I like that. Yeah. Well, I don't think most people outside of 1 or 2 minutes in their life worry about it because it's not possible, right?
Exactly.
Although we didn't know we could get ancient DNA not too long ago, right? So.
Oh, we can push it back pretty far. I think if you're in conditions which are very good at preserving ancient DNA, think of the permafrost, you can go back maybe 1 million years. I think after that there's no hope, but 1 million years is still a long time.
Agree.
This is a great story, isn't it, Nels?
Yeah, and a natural podcaster, I would say. So if you guys wanted to do a 2 Vinny podcast, it seems like a viable situation.
Sort of the title writes itself, the Vinnies.
The Vinnies. The Vinnies, yeah. Yeah, well, Nels, we can't exclude you, so we'll have to come up.
I appreciate it.
We'll have to come up with another title. You got anything else, Nels? You want to move on?
Yeah, I think that covers it. So,
wait, one more thing, of course.
Oh.
Well, what's next, Vinny?
Oh, yeah.
So the effect sizes of all of these things are relatively small. So you can measure the sort of contribution that this extra copy of LIF makes to the apoptotic response, and you can measure the contribution that those extra copies of P53 make to the apoptotic response. And they're small, on the order of, a few percent, which indicates actually that however elephants evolved it, these particular genes are only a small part of the story, which means we still have to explain all of the other parts of the story. So we're trying to do that now.
Yeah. I also like the experiment with zebrafish. I'm looking forward to that. That'll be cool.
Yeah, that'll be fun.
You still there, guys?
And we're also moving on to whales and bats because they're pretty interesting too.
Yeah, bats are really cool with all that. But they have so many antioxidant genes.
Yeah.
And that sort of thing. We're interested, or virologists are interested because they have so many viruses in them.
Yeah, that terrifies me. That's why I work with their cells.
Yeah. Nels, you still there? He left. It's you and me, Vinny.
See, it's the Vinnies.
I'll get him
back. We don't
back.
need Nels.
Yeah, I just got cut off. I guess they're on to me.
It was
the authorities are.
It was 2 Vinnies for like a minute there.
The podcast actually happened.
Yeah, just us. That's right.
Yeah, sorry about that. I think my Ethernet connected internet went south, and so now I'm on the Wi-Fi connection. It seems to be working.
All right. Well, let's do some picks before you leave us forever.
I've already been replaced, upgraded, just like LIF, the parental ancestral LIF gene.
That's right.
LIF6 has come in to refunctionalize and replace me.
That's right.
Yeah. So, Vinny, for your new podcast, we also do this science picks of the week. I've got one. Vincent will do one, and then if anything comes to mind to you, just off the top of your head, just sort of a internet tidbit or something interesting that you've seen, feel free to do a guest pick of the week if the spirit moves you. So, Vincent, my pick of the week is a series of short videos done by Ed Young on Facebook called Animalism. And he's now, I was just looking today, there's 5 episodes. This is, so Ed is obviously a really famous science writer who's done things. His latest book, I think, is I Contain Multitudes on microbiomes. He's also a prolific writer at The Atlantic. And so this is an initiative through The Atlantic where he's been posting these really artful and interesting short videos on different animal behaviors. And so I've watched the one on how animals see the world, looking at the diversity of sort of eye structures and strategies for vision or perception. There's another one that just got posted recently on the naked mole rats and how they can survive without oxygen for something like 20 minutes.
And they never get cancer.
And I was gonna ask you about that, Vinny. This is a small critter that doesn't get cancer, so maybe
never
it's
gets
already
cancer.
Yeah. What's going on with that?
Oh, they're crazy.
Yeah, maybe the topic of another episode. So, but the reason I wanted to highlight this is just that I think, for me at least, this sort of combination of the art with the scientific story, sort of told in interesting and compelling ways, is just really inspirational. And so I would recommend checking out I guess it's on season 1 now, 5 episodes, all of them very well done.
Who's making these? Do you know? The Atlantic doing it?
Yeah, it's a good question. So I think that's right. We might have done or kind of been involved in a prototype of this. So when Ed Chung from my lab, who's co-mentored with Cedric Fichat, he had a paper that came out on how endogenous retroviral elements can wire up the interferon regulation. Ed Young covered that paper in the Atlantic, which is really cool. But he also, it didn't really show up, but they made, with the Atlantic, they made a short video, about a 3-minute video, as maybe like a pilot run of this. Maybe I'll see if I can dig up that link. I think it's really cool too, but that was never sort of put together or packaged. And so maybe this is sort of a result of that, that they kind of reloaded and got a theme together, in this case for season 1 on animalism, all these different animal behaviors. And so it looks like it's advancing in a cool way. But yeah, I think a collaboration between the Atlantic and with his position there. Are
they just putting it on Facebook? It's not on YouTube?
As far as I know, it's on, it's a Facebook channel. So, and I really like this idea actually and I think, I got this from you, Vincent, was that, we already have these kind of social networks that are often outside of just science on Facebook, family and friends. And so what a great place to do outreach.
Mm-hmm. Yeah, you get a lot of those people. That's why I post everything there as well, even though I don't do much on Facebook, but I like doing it. The video is great, of course. These are really nice. They're cartoony and short, and I think that's a great way to reach a lot of people and teach them something.
Yeah. Yep. Ed is really an impressive communicator.
It's nice.
Yeah. How about you, Vincent? What's your science pick of the week?
Well, I have an article from NASA, which is really cool. Voyager 1 fires up thrusters after 37 years.
Wow.
So Voyager 1 spacecraft has been up there 37 years. I guess the thrusters, it's been flying for 40 years, but these thrusters, of course, it's way past our solar system.
Mm-hmm. Was this the one that was it last year that it kind of reached the boundary and went across?
Yeah, I think so.
Remember that right?
Think so, yeah.
Mm-hmm.
It's 13 billion miles away from Earth. And they haven't used these. So it still can communicate with Earth, right? And it has to reorient itself to do that. And they use these little thrusters that fire millisecond-long puffs just to rotate it so the antenna points at Earth. And they had 4 backup thrusters they haven't used since 1980. So they said, let's try and use it, and we could get more information for a couple more years, because I guess it was going to run out, or the other ones were going to stop working, run out with whatever it is that puffs out, And so they sent some commands, and, a day later it responded, and they're like, holy crap, it still works.
Wow.
I just think it's amazing that this thing was built so well that it's still working. And
yet Nels can't keep an internet connection.
Yeah,
that's right. That's right.
Guilty as charged. Yeah.
It's just a testament to NASA. I think they do, with a really small budget, half of that of NIH.
Right.
They do amazing stuff, and they make amazing hardware. And this is just, just think, this thing is way out there, and it's still responding. So I just think it's a cool story.
Agree. Really breathtaking.
You can do that, yeah.
Yep. How about you, Vinny? Anything come to mind on your end?
Yeah, sure, but it has nothing, to do with science.
It's okay.
Really.
That's good.
So there was a series of clips going around on Twitter called "What if animals were round?" Basically, they're animations of what life would be like if animals were round. So basically, round with little tiny arms, and they're just really hilarious. So, for example, there was one of a crocodile trying to attack a flamingo, and it pops up from the surface of the water, but because it's round, its arms can't actually help it swim, so it just bobs around like a balloon sitting in the water. They're very funny. I guess there is a little bit of science there, because it goes to show you about things like biomechanics, and if all organisms can't be the same shape because they have to adapt to their environments. So I can make it sciencey, but they're funny. And if you have a moment, just Google "What if animals were round?" and watch the clips.
Yeah, they have a website. It's called Rollin' Wild, right?
Oh yeah, they do.
That's it, right? Yeah, that's where the clips reside. Nels, are you still there? He went away again.
Oh, see, technology these days.
NASA.
It's no NASA thruster.
Hey, Nels.
Back again.
And Vinny said, NASA can do this, but you can't.
Well, some people think of Salt Lake as being about 13 billion miles away. So,
all right. Yeah. Well, we should wrap it up. I found the website for round animals. So
that sounds amazing. I can't wait to take a look under the hood.
Cool.
That sounds interesting. Yeah.
Pretty funny.
All right. This is TWiEVO 26. You can find it at Apple Podcasts, microbe.tv/TWiEVO. Of course, if you have a favorite podcast player, you just search for TWiEVO, you'll find it. Please subscribe so you get every episode. And if you like what we do, consider supporting us financially. Microbe.tv/contribute. And questions and comments, TWiEVO@microbe.tv. Our guest today has been Vinny Lynch from the University of Chicago. Thanks, Vinny.
Thanks, Vinny. This was fun.
No problem.
What's your handle on Twitter?
So it was Vin J. Lynch, but I just turned it into TWiEVO Med.
That's all one word?
All one word.
TWiEVO Med.
What was the motivation for doing the change?
Oh, I was just playing around.
Cool.
You like Chicago?
I think this one's more sciencey.
You like Chicago, Vinny?
Yeah, I do. Chicago's a great city.
Yeah.
Actually, yeah. So I have this love-hate relationship with cities. I love cities because of culture and food, but I like green and outside.
Yeah, sure.
And where I live in Hyde Park in Chicago, there's a little bit of both. There's no mountains because there's no topography of any kind in Chicago, but you can get out along the lakefront within about a mile, so it's kind of nice.
Yeah, I like Chicago. It's a good city.
Me too. Totally agree.
It's a little more manageable than New York, I think. And
oh, absolutely.
It's got more space. They laid it out really well, right?
Oh, yeah, they had a chance to once the fire came through.
Yeah, that's right. Nels Elde's at cellvolution.org. And on Twitter, he is L Early Bird. Thanks, Nels.
Yeah, thank you, Vincent. Great to be with two Vinnies. I've really enjoyed it.
It was kind of awesome.
That was a great topic, too. I love it. It's just so cool.
Yeah, exciting work.
So good choice, Nels.
Thanks, Nels.
Pleasure. Thank you. Great to be with you guys.
I'm Vincent Racaniello. You can find me at virology.ws. I am PROFVRR on Twitter. The music you hear on TWiEVO is by Trampled by Turtles. You can find them at trampledbyturtles.com. Been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.